Good afternoon. We're just going to wait another minute or two for people to filter in to the Zoom webinar. But while you are waiting, we have this poll here for you to take. So there's two ways you can submit your answers to the poll. The first way, there should be a link in the Zoom chat where you can answer the poll. The other way is through text message, which you can see on my screen. There's some instructions there on how to text. So you can go ahead and do that. So I'll give you guys a minute or two to respond to the poll. All right, great. It looks like you guys are responding to it, so that's perfect. All right. Looks like a lot of you guys have taken a COVID test before. So if you have just entered, we're just taking a poll. On the screen, it just has some instructions on how to submit your answers to the poll. There's also a link in the chat as well. All right, great. It looks like a lot of people have taken a COVID test. So that's wonderful. So we hopefully you guys can learn a little bit more about what we do at the Diagnostics Accelerator on COVID testing. All right, cool, 50-50. So half of you haven't and half of you have. Oh, look, it's changing. Okay, great. So we can go ahead and start. So thank you all for joining us for Inside a Lab, putting COVID to the diagnostics test. This is part of the Cambridge Science Festival event. So we're super excited for you guys to be joining us here today. Before we start, just wanted also to give you a couple of sort of housekeeping things. The first thing is that we do have closed captioning available. So if you're interested in using closed captioning, you can do that by clicking the closed caption button in the Zoom bar below to view the closed captioning. The event is also being recorded and will be available later for viewing. We can send an email out when that is available. So I'm just going to pass it over to David Walt, who's the co-leader of the MGB Center for COVID Innovation. He'll talk a little bit more about what we do in the center and also a little bit about the work that he does as well. So I'm just going to pass it over to David to talk a little bit about himself.
Thanks, Sarah. Hi, my name is David Walt, and I'm a professor at Harvard Medical School, and I'm also a principal investigator at Brigham and Women's Hospital, and that means I run a research lab. So how did I become a scientist? Well, when I was young, I just loved being outdoors and exploring nature, and I also had a chemistry set, and I did experiments in my basement. I also had a workbench where I loved taking apart old radios and other electronics equipment, but I never put them back together. I was just curious about how things worked. When I went to college, I started working in a lab in my freshman year, and I became hooked on science. I realized that the experiments that I was doing would enable me to learn things, and I'd be the first person ever to know them. Imagine that. Of all the billions of people in the world, you know something that no one else does because you discovered it first. Now at Brigham and Women's Hospital, I run a large laboratory where we develop new diagnostics technologies. And many of these projects involve ways to measure molecules at really, really low concentrations. We measure things called molecules. I'm sure you've heard of this, particularly certain proteins, which are sort of the workhorse of the human body. And these proteins have never been detected previously because they've never been able to measure, scientists have never been able to measure these things in the blood or in various other fluids of the body at the concentrations that we can measure them. So our hope is that these proteins will be able to help us detect diseases such as early detection of cancer and Alzheimer's and Parkinson's diseases, so people can be treated before the disease progresses to more serious stages. Let me tell you a little bit about the center. When the pandemic first hit Boston last March, we started the Mass General Brigham Center for COVID Innovation, of which I'm the co-director, as Sarah mentioned. We helped figure out ways to produce and reuse protective clothing and masks that were needed to protect the healthcare workers and patients because they were in short supply at the beginning of the pandemic. We worked on quickly developing new diagnostic tests and also finding effective drugs to prevent patients from getting seriously ill. Our work is continuing to have an impact both in the local communities, but also all over the world, as you'll hear a little bit later from another Sarah, who's going to talk about the diagnostics accelerator. So to get you started on the lab visit, I'd like to introduce you to Dr. Sarah Suleiman, who has been running the diagnostics lab for the center. Sarah, take it away.
Thank you so much, David. I'm so excited to be here today and to talk to you about the Diagnostics Accelerator. So just to give you a little bit about myself, I'm an immunologist by training. So immunology is the science of studying how your immune system can fight infection. So obviously when the COVID pandemic started, that was the right time for me to use that skill set for fighting infection. Just to give you a little bit of background about my research, I work on tuberculosis. Tuberculosis is very similar to COVID in that it's an infectious disease of the lung. And just like COVID, a lot of people actually globally get infected with the bacteria that causes tuberculosis. But then some people get severe disease, some people get mild infection, some people get no symptoms at all. Actually, the majority of people who get infected never get any symptoms. So my research looks at what is it about those people who progress to active TB disease that is different? Are they expressing different genes? Can we use diagnostics that actually predict who will be on their way to developing severe disease? So as you can see, diagnostics are very important in predicting different stages of infection and disease. And as David mentioned, when the pandemic started, the hospital started this COVID Innovation Center with a lot of focus on diagnostics. And this is important because a lot of technologies came into play, but we needed to know how accurate they were. Are they actually picking up all the people who have the infection? Or are we missing some people? Are there false negatives? And likewise, are we overdiagnosing people, and maybe people who didn't have the infection are coming up as positive in these tests? So that was the purpose of the Diagnostics Accelerator Lab. So what we do is evaluate these technologies before we can use them in the hospital setting or in the community at large. We know now that there are some tests that are going to be used in community testing sites or for home use. So before we can basically put our stamp of approval on these tests, we need to make sure that they're accurate and they actually have the performance that we expect them to have. And I became a scientist because I want to understand the why and how of how diseases happen and how we can use that knowledge basically to have better interventions and better vaccines and treatments, etc. And I'm most excited about the team of amazing young scientists who are the technicians in this lab. And I would like to introduce them in the following video that Sarah will play for you.
Hi everybody, my name is Marlene de Leon. I am a research technician here at the MGB COVID Innovation Center. And so we evaluate different direct-to-consumer platforms that can help like predict or test accurately whether a person has COVID or not. So we are doing different like research related to immunology, so we can understand better how COVID, can be predicted. So, and I also work as a research engineering technician at the Wellman Center, where we do similar work as we do in the COVID center. We do diagnostics, but in that case for the gastrointestinal tract, and we fabricate different devices that can help detect diseases and prevent diseases to progress.
Hi, my name is Sujata Chalise and I'm a research assistant at Center of COVID Innovation. We basically work in validation of diagnostic devices for COVID-19. I'm from this beautiful South Asian country, Nepal, which has been greatly affected by COVID-19, not just in terms of death and positive cases, but also in terms of economic crisis, poverty, unemployment, and people dying of hunger, which is very saddening for me. But the fact that I'm a part of this amazing project, which is trying to contain the ongoing crisis, it gives me immense satisfaction and hope. It makes me feel empowered.
Hey, I'm Zach Anadero, and I am actually a cell molecular bio undergrad at Northeastern University, as well as a lab tech. While I'm here, I usually end up working either bench side or in some slight project management to make sure everything runs smoothly. But most of the time it's over here evaluating different COVID tests and technologies.
Hi, my name is Sally Zoe, and I'm a research assistant in the COVID-19 DA lab. And as a research assistant, I help run COVID-19 samples on various diagnostic platforms. And I like science because I like learning how the body works.
All right, so I'll take you all through the Diagnostics Accelerator Lab. So as you can see on the left here, we have a freezer where we keep our samples because a lot of them have to be kept at really cold temperatures so that they don't lose performance basically on the diagnostic test. And here we have a safety shower. Hopefully there's no chemical or chemical spills or anything that would require us to use it, but it's here just in case as a biosafety measure. And as you can see here, we have the PPE or personal protective equipment, which we will talk a little bit more about in just a couple of minutes. And this is our COVID zone. You can see there's a line on the floor which separates the non-COVID zone from the COVID zone. So if you need to go to the other area, you need to wear additional layers of PPE to make sure that you're protected once you're actually handling samples. So you can see two of my favorite people here, as Marlene and Sujata, are both running diagnostics tests and evaluating whether the performance of these tests is what we expect them to be. All right.
Thank you, guys. It's an honor for us that you guys can be here with us, sharing what we love to do every day to help in our best way to people who as we know, might contract this disease. So I'm Smarling, as Sarah mentioned. I'm a chemical engineer and I work in Guillermo Sterni's lab. And it's just been one of the most amazing opportunities for me, to be here at MGH, because I think there's a lot of opportunities to grow as a scientist. And for me, as in a way, I'm a young scientist. it was very important to me to be able to use my training and what I know about chemistry and experiments to help in the best way possible during this pandemic. So I'm very happy to share with you guys. I really love like this team and how all the professors and researchers have gathered together so to put the center and run it. And I appreciate a lot Sarah and David and Gary and all the other people. And I also wanted to tell you guys, since I know there are many kids in the call, so I wanted to tell you what a scientist looks like. And so in my case, I started when I was in high school. And something that was very curious to me was just watching a video of someone showing how in the future we can maybe have like materials or different chemicals that can help us change the colors of our t-shirts, or like how we can develop like just new ideas that maybe we see in the movies and we say like, oh, I don't think it's possible. So for me, science is that lens in which I can see just how we can create many things that can help us like move forward, or just like even go to space, or like cure patients, like cure diseases. So here at the COVID Center, I really love what we do here, because like at the beginning, I didn't know how I could help, and I was very scared, as all of you are, and probably were like a year ago. So in my lab at Turney's lab, they gave me the opportunity to join the diagnostic accelerator and just help whether I can. So I started as a volunteer, and then I learned the experiments and how to analyze the data and all that. I had the opportunity to supervise other volunteers and just help the heavy load that we had of testing many different samples and trying to understand the virus. So that was very nice, because like a year from now, yeah, we will think about this and how we help many people and how we could understand how the different devices we test are accurate, or whether we can improve them so we can diagnose ourselves in a very way. And finally, I want to just show, as Sarah mentioned, like PPE or personal protective equipment is very important in science, because it's like I think about it also like maybe as astronauts wear a spacesuit, we need something that can protect us depending what we are doing, right? So here at the COVID Center, we have different protection, because we work like as you can see here, that's like biosafety level 2. So that's something that the Center for Disease Control has developed, so we can know, like depending on the specimens or type of microbes we are dealing, like how we can protect ourselves, so we can safely do our experiments and help many communities. And as you can see, Sujata here, my dearest colleague, she is performing like different experiments. She's wearing the PPE, and we will show you later like how everything comes together, and we will show you how we can put our PPE in the next video in which Zach will be doing the donning. Thank you.
Hi, everybody.
All right. So next, we will actually go through an experiment that Zach performed in the lab, and it's an evaluation of a lateral flow assay. So these are devices that look like a pregnancy test that test whether or not you have antibodies against the virus. And the center has actually evaluated more than 20 different lateral flow assays from different manufacturers. So let's watch Zach. Okay, so let's walk through an experiment for testing for COVID antibodies. So Zach right now is cleaning the bench and using ethanol to wipe off the different surfaces, as well as the plexiglass, which you see there. So that forms a shield where he can actually analyze the samples behind the shield, so that reduces the risk of exposure and infection with samples that are potentially contaminated with SARS-CoV-2, which is the virus that causes COVID. So what he's doing now is just after wearing the PPE, or the personal protective equipment, he's taking out the lateral flow assay. So these are assays that you can use to detect antibodies. So if your body has generated immune responses against COVID. So you can see that in the box on the left of the plexiglass shield. He's taking out one of these lateral flow assays, which basically look like a pregnancy test. So if you have a positive result, you will see a band. And if you don't, then you don't see a band. So the band will indicate whether or not you were previously exposed to COVID, which will then tell your immune system basically to generate antibodies. So he's now looking over the paperwork, making sure that we have exactly the right sample number. This is a research lab, so we don't report the results back to patients, but of course, we want to make sure that the results that we're recording are accurate and correspond to the right samples. So what he's doing right now is just getting the sample to room temperature because it was frozen. And he's using a pipette, so that allows you to take out exactly the right volume of sample so that you can add it to the lateral flow assay. All right, so now that the sample is thawed and it is at room temperature, he will be adding it to the LFA, or the lateral flow assay. All right. Now he's ready to go. All right, so first he's taking out the lateral flow assay out of the package and adjusting the pipette so that it takes exactly 10 microliters of the sample, so he can add it to the lateral flow assay. All right. So this is the package. Everything now is at room temperature. Double checking the instructions on the package insert just to make sure that we're following exactly the instructions for using this device. Okay, now Zach is taking out 10 microliters using the pipette from the sample and then adding it onto the lateral flow assay. It's a very simple and straightforward way to analyze whether your body has generated antibodies against SARS-CoV-2. SARS-CoV-2 stands for Severe Acute Respiratory Syndrome of the Coronavirus 2, and that's actually the technical name for the virus that causes COVID-19. So after adding the sample, he's just basically adding 2 drops of the buffer that comes with the kit, and that allows it to flow through the assay. That's why it's called a lateral flow assay. And then it interacts with the antigen, which is on the device. So if it is present, then you will see a positive band. So we can take a closer look to the lateral flow assay right now. You can see that there are 3 different bands. One band is a control band, so that tells you that you have a valid assay and there are no manufacturing issues with it. And then there are two other bands. So one says M, which means IgM or immunoglobulin M, and one says G, so immunoglobulin G. And those are two different types of antibodies. So this sample clearly comes from a positive sample. All right, so I see a number of questions. David, would you like to take over moderating the questions?
Yeah, sure, that'd be great. So we actually have some great questions already, and I encourage people, if you are putting a question in and you're not an adult, please put your age. We'd like to know the age of some of the attendees. So I guess the first question is really for one of the technicians. So Sarah, I'll let you field this one, but was it scary to do the work before the vaccine? I think that's a great question.
Yeah, I think that's an absolutely fantastic question. And the short answer is yes, but that's exactly why we had all these protocols in place. We had a very detailed biosafety protocol, which tells us exactly how many times we needed to clean different surfaces, what types of PPE we wear, what zones in the lab we can actually analyze these samples in. And also we had to deactivate some of the samples. So if you have for the 50% of you that did a COVID test before, you probably had to do a swab. So we actually had to deactivate those samples and heat them up so that the virus is dead before we analyze it. So we took all kinds of precautions to make sure that the samples are not infectious. And of course, as soon as the vaccine was available, we all got vaccinated as well.
Okay, another question: how many tests has the lab done?
Yeah, quite a lot actually. So we've done 20 lateral flow assays. So those are for the antibodies. We analyzed 2 PCR assays. So PCR stands for polymerase chain reaction. So those would diagnose active infection and test whether you have genetic material from the virus. And we've analyzed quite a few antigen tests. So those are also like the lateral flow assays, but then instead of looking at the antibodies, you look at whether the protein from the virus is present. So we analyzed those both in the lab and also in a community testing site in Western Massachusetts.
Okay, well, Elena O'Malley, who characterizes herself as a tween, asked the question of, does COVID transfer to objects sufficiently to infect someone?
That's an excellent question. There was quite a lot of debate at the beginning of the pandemic on how long the virus would actually stay on different surfaces. The virus is transmitted aerosolly, so mainly through particles in the air. But because there is a risk of transferring the virus when you cross-touch surfaces that are possibly infected with the virus, that's why we have very detailed protocols for cleaning the surfaces many times a day and making sure that there's no transfer of infection. The short answer is probably, but probably not to the extent that we were scared of at the beginning of the pandemic.
Yeah, I think that also gets to one of the earlier questions where if you spilled it and it was a live virus, you know, from an NP swab or from an infected patient, I think there's a higher risk than just transferring from inanimate surfaces, you know, like from groceries or something like that. Constance Richford asks a question that I will answer. Can the test differentiate between antibodies from past sickness versus antibodies from the vaccine? And can it tell which vaccine is which? So this is a really great question. And the answer is that some of the tests can and some of the tests can't. And the reason for this is that the different types of antibody tests look for the antibodies to different kinds of proteins. And there's different proteins that the virus produces. I think you all know about the spike protein. And the spike protein, that's this protein that sits on the virus and decorates the membrane of the virus to make it look sort of like a corona. which is what gives it its name, that particular protein is what all the vaccines are targeted toward. So the antibodies that are produced against the spike protein, one cannot distinguish between antibodies that are produced from natural infection versus the vaccine if you're just testing for the spike protein. But some of the antibody tests test something called the nucleocapsid protein, which is a protein that's inside the virus that if you get an infection from the virus, then you will produce antibodies to both the spike and this nucleocapsid protein. And for those antibody tests that test for that particular protein, then you can tell if somebody was naturally infected or just developed antibodies to the vaccine. The second part of your question is actually one that I don't think anybody really can tell yet, but it appears that it's more of sort of a yes, you've had the vaccine, no, you have not had the vaccine. The different vaccines actually elicit very similar kind of immune responses. So the answer is probably no. Okay, let's see. What advice do you have for high schooler interested in pursuing healthcare and biomedical science careers? Sarah, I don't know if you want to take that first. I'm certainly happy to answer that as well.
Yeah, I think we can both give our perspectives. I would say definitely do it. If there's anything we learned from during the pandemic is access to accurate information. And being a scientist myself, I was actually able to understand what's accurate and what's misinformation, and develop the skills to decipher that. So if you do want to become a scientist, I suggest starting off with reaching out to different labs, either for volunteering or for summer internships. There's quite a lot of opportunities, especially in Massachusetts. That would give you an idea about what a research lab looks like, and or what the day-to-day life of a scientist actually looks like, and also develop the critical thinking skills that are necessary. And during your undergrad, you can diversify what types of courses that you take that would give you exposure to different areas of science. And then you can make a decision if you want to pursue additional research to go into graduate school and pursue a master's and a PhD if research is what you want to do. David, what are your thoughts?
I agree with everything you said, Sarah. I think that the only thing that I would add is that I don't think it really matters sort of what field you go into. If you're interested in college, I think you should just follow what you're really interested in. Obviously, if you're interested in science, you'll probably pursue a science major. But what I would say is that whether it's physics or chemistry or biology or some sort of technology, engineering, I don't think it really matters. There's very good ways, very good paths to go into health science and the medical field from all of those different fields, and we need everybody. Engineers make things that other people can't, and biologists can study and learn things that engineers can't, and chemists study different things. And so again, I think it's really what you're passionate about, what you're interested in. I think you can enter it from any of these different areas. Sophia Shepard, who's 13 years old, has the amount of protection that you use in the lab lessened after the release of the vaccine?
Excellent question, Sophia. So actually, no, we kept using the exact same levels of PPE. And the reason for this is because we want to make sure that when you're dealing with samples where you have a higher concentration of the virus than you would get, say, for example, for eating at a restaurant or, being in close proximity to someone who has COVID, we want to make sure that the risk of exposure is as low as possible. with techniques like PCR, when you amplify the genetic material or etc., we want to make sure that as much as possible, there's absolute protection for the technicians. And maybe those guidelines will change in the future, but as of now, we're maintaining the same level of protection that we had at the beginning.
This is a great question. It's an anonymous attendee. Do you test for COVID-19 too, or is the focus on antibodies? And what role does the protein amplification play in testing for COVID? What factors are considered to inform the degree to which the protein should be amplified? I think just answering the first part of that, do you test for things other than antibodies?
Yeah, so when we test for antigens, that means we're testing for the presence of proteins that are made by the virus. When we test for using molecular tests, so the PCR test that I talked about earlier, we're looking for the genetic materials that come from the test. So they all have different, what we call use cases. They have different reasons for using them. I lost your question. What was the second part of the question?
It has to do with the way that the protein gets amplified.
Yeah, so we have the sequences of the virus, and different tests would amplify different regions of the virus, and each developer can choose different regions. What we want to target as much as possible is the areas that have the least amount of mutation. So you want it to be conserved in different samples so that the instrument can accurately detect the presence of the virus, regardless of whether new mutations happened or not. Usually they target the N region, so that's the nucleocapsid. So that's present in the virus, but not in the vaccine. Just on the question before on antibodies that distinguish vaccine-induced immunity versus natural infection, we can use the nucleocapsid for that because the vaccines target the spike region of the virus.
Another question is, if you weren't a scientist, what would you be doing instead?
Is that for me?
We can both answer.
Yeah, I've always wanted to be a scientist. I think an alternative career would have been writing. I really enjoy writing, but at this stage in my career, I actually do a lot more scientific writing. So I feel like both worlds have finally merged, which is great.
Well, I guess I have the same answer. I always, wanted to be a scientist or was interested in science, and not sure how to answer the question. What were you researching before COVID?
Yeah, so I mentioned at the beginning I was working on tuberculosis, it's very similar to COVID in a lot of aspects because it's an infection of the lung, and it also has the different outcomes that make it biologically interesting. What is it about the different genes or different immune responses by different people that make someone get severe disease versus no symptoms at all? So I've been working on TB for the past 8 years. And just to echo what David said earlier, there's different paths to science. I was actually working on leukemia and cancer before I started working on TB, and then my interest switched. So that tends to happen when you have a career in science.
Sarah, I think this is a great question from Mary Cat Boyette. Will we eventually be able to use the test like what he did in the experiment at home? I think we know the answer to that.
Yeah. So the FDA, or the Food and Drug Administration, actually now authorized a few of these tests to be used at home. So that happened on March 30th. Obviously, I keep track of these things. So those are antigen tests. So they tell you if you have an active infection by detecting the presence of proteins from the virus. So they're lateral flow assays, but not for antibodies. They actually detect the actual infection itself.
Okay, we have a recent question from Eureka Tarui. What, if any, differences have you noticed in antibody testing results across different socioeconomic, demographic, and medical backgrounds? I'm 16 years old, and that's a great question. We know that there are significant disparities both in the access to testing in different communities, as well as the prevalence in different communities. But Sarah has just carried out a study in Holyoke. So Sarah, do you want to talk a little bit about that?
Yeah, so there's definitely ethnic disparities in how the infection load is distributed, especially in Massachusetts. So the way to know that is something called the sero survey or seroprevalence study, where you test out of the population how many people have been exposed to COVID before. So unfortunately, Black and Brown communities have a higher prevalence than white counterparts, for example. And also, depending on the time of the year, you will see different rates of infection. So immediately after the holidays, the testing positivity rate, so then the percentage of people that turned out to be positive when they came to test were up to 10% actually, which is really high. And now we're hovering around 1 to 2%. So that's really good news. That means that even when you suspect exposure, the chance that you have COVID right now is, or that you have infection with the virus is much lower.
And then our last question, Sarah and I had alluded to this earlier about, and this again comes from an anonymous attendee, who's using these tests in the US and around the world? And maybe you'd like to talk briefly about what we're doing with some of our global sites.
Yeah, that's an excellent question. And I saw there was another question about the difference between PCR and antigen. So PCR tests require big instruments, usually take a lot of time to get the actual genetic material from the virus and amplify it. So that needs access to a central lab where people who are trained can actually run these tests. When you go to rural communities or resource constrained settings, like low and middle income countries, for example, they rely more on rapid tests. So those are lateral flow assays that detect the actual infection. So that allows for more access to testing than we would with PCR by itself. So we're working with Kenya, Malawi, Botswana, and Bangladesh right now to evaluate how these rapid tests would work and compare them to the PCR, which is considered the gold standard. So that's what we use to benchmark whether a sample is positive or negative. That decentralized access to rapid tests would allow more people to get testing in resource constrained settings or when they don't have access to central labs.
Okay, I think that brings us to the end of the questions. There's still some very good questions, but we just don't have time to answer them all. So I'll turn it back to the other Sarah to wrap up and close the event. Sarah.
All right, thank you guys. Let me just see if I can. All right, thank you guys for joining us today.